This is my demo for bicoid action in Drosophila. First I have here an unfertilized egg of Drosophila. This is the egg cell nucleus, and this is the egg cell. These are the nurse cells, and these are the nurse cell nucleuses. The mRNA molecule that specifies the anterior-posterior axis of the embryo is transcribed from an egg polarity gene which codes for a protein called Bicoid. Bicoid is a maternal effect gene that codes for a protein responsible for specifying the anterior end in Drosophila. It is a morphogen and is seen to be concentrated in cells at the anterior end of the embryo. The nurse cell DNA is where the bicoid mRNA is created. Transcription factors within the DNA is what determines the function of the mRNA. The bicoid mRNA will thus exit the nucleus through nuclear pores. Once the bicoid mRNA is transcribed, it is deposited into the Drosophila egg asymmetrically. More bicoid mRNA is deposited at one end of the egg than the other. Once the egg is fertilized from the sperm, the bicoid mRNA is translated to bicoid proteins, which is done with the help of egg ribosomes. The bicoid protein then diffuses from the anterior end toward the posterior. This results in a gradient of protein within the early embryo, with the highest concentration at the anterior end. The bicoid protein gradient establishes the polarity of the embryo along the anterior-posterior axis. Gradients of substances called morphogens establish an embryo's axis and other features of its form. The head and thorax will develop in the zones of high bicoid concentration. And this concludes my demo.